Good morning or good afternoon, everyone. My name is Marie Latulippe, and I'm the Director of Science Programs at IFINS. And we're very pleased to offer you this webinar today, which is focused on the taste modality of sweetness. And for those of you that might be new to IFINS, we are the Institute for the Advancement of Food and Nutrition Sciences. And we're a nonprofit organization that advances food and nutrition science for the benefit of public health. And the way we do that is we bring together scientists from different sectors to decide what their common interests are and then activate research programs around those interests. And on the left of the slide here, you see the core values of IFINS. You can go back one slide. And those include scientific integrity and transparency, to which we have a very strong commitment. And the next slide illustrates that this webinar is approved for one continuing education credit by the Commission on Dietetic Registration. So after the webinar, we will be providing access to that certificate to all the participants today. Before we start, we just wanted to make a quick note on logistics. For those of you that have joined us for previous webinars, you're familiar with this platform, but basically you should see something like that control panel that you see on the slide here. And there's going to be a line that says questions. Please go ahead and type your questions in that box throughout the webinar. And what we'll be doing is taking questions at the end for all of the speakers. So with that, I'm extremely pleased to turn the program over to Ann Colston. And Ann will be serving as our moderator today. Ann has a more than 25-year history of clinical research, and she's provided nutrition consultation to the food and healthcare industry, public relations firms, as well as internet companies. And Ann, if you'd like to join us with your webcam, you're welcome to. Ann is also past president of the Academy of Nutrition and Dietetics, and she's also been recognized by the A&D Foundation for excellence in the practice of clinical nutrition and also in the practice of research. So Ann, we are very pleased to turn the platform over to you today.
Thank you. We're excited to bring you today's program, which focuses on the role of sweetness in the diet, as well as presenting some new data on the use of low and no calorie sweeteners and quality of life. Let's get right to the program by introducing our first speaker, Nancy Rawson. Dr. Rawson is the acting director and president of Monell Chemical Senses Center, a nonprofit research institute dedicated to the study of taste, smell, and related senses. Dr. Rawson holds multiple patents, has authored over 100 publications, is active in the Association for Women in Science, and serves on the boards of the Smell and Taste Association of North America and the Institute for the Advancement of Food and Nutrition Sciences. We welcome Dr. Rawson, who will begin speaking about the biology of sweet taste. Dr. Rawson?
Thank you, Ann. It's a pleasure to be here talking to you all this morning or afternoon, as the case may be, about sweetness and the biological drive for sweet taste. So, I think it's important to really go back and think about, why do we taste? What is the role of this sense? And we know that every species has to solve the fundamental problem of obtaining sufficient nutrients and avoiding being poisoned. A species' taste sensitivity and preference is co-adapted to its ecological niche. For instance, we know that the obligate carnivore, the cat, does not have a functional receptor for sweet taste. But in fact, the genes for sweet taste receptors evolved over 400 million years ago, before the first vertebrate even walked on land. And you can see this image here of what that creature might have looked like that first evolved this ability to detect sweet compounds. So this sense really evolved as a nutrient signal so that we can find the nutrients that we need in our environment. So what's important to understand is that in order to recognize these nutrients and to remember that this is where we can get nutritional value. There's the senses of taste and smell are the basis for flavor-based learning. So taste and smell are the cues that really enable us to learn about the health consequences of the things that we consume. Are these things going to be toxic? And often bitterness is a signal for toxicity. Or are they going to be nutritionally valuable? Things like sweet or sugar or fat or protein. And so we have positive or negative hedonic responses, and these responses shape our behavior in order to ensure that our nutritional needs are met. Sweetness in particular is a signal for the presence of calories. And sweet liking, it's very clear, is present at birth in all cultures and even in primates, other primate species. So really, you can understand why it's so difficult to redirect the effects of this survival-driven biology and learning. And in fact, this has been attempted because we knew that with salt preference, if you go on a diet that's low in salt for some period of time, your ideal or your optimal preferred concentration of salt does decrease. So you can actually shift your preference. So can we shift preference for sweetness? And this is one small study that was done. There's two larger studies currently in progress to looking at this question in more depth, and it'll be exciting to see the data coming out from those studies. But for the moment, this is really the only study that addressed this by Paul Wise and colleagues, where they reduced sugar intake by 40% for 3 months. And they looked at preference for a couple of different foods, and I'm showing here the results for the sweet pudding. And what you can see is that really there was no change in the perception of intensity over the first 2 months of the lower sugar diet, but the intensity was perceived as more intense for the low sugar pudding. By after 3 months of this low sugar diet. If you look, they did then at this point, they went back on their normal diet, and you can see that intensity started to come back towards where it was before being on the diet. So it was a temporary effect. The other thing, in addition to looking at whether the subjects perceived the sweetness as more intense, they asked about their liking. And unfortunately, there was no effect on the preferred level of sugar in the pudding. So even though at this point they perceived the sweetness as more intense, when they tested their preference for pudding with different levels of sugar, they just preferred the one that was more intense, that was the same concentration as in the beginning. So it's clearly very hard to shift this preference. However, the studies that are in progress are looking more comprehensively at things like equating the amount of sweetness in the diets of these individuals. In this particular study, it was not a fully controlled dietary study where they knew all of the foods that the individuals were eating. So it was more of a pilot study. So to start understanding how we can attack this problem, we really need to think about how sweet taste works. And the oral taste cells are the initial chemosensors of the alimentary tract. So, you know, they are present in the tongue, but they're also present throughout the gastrointestinal tract, and that's an important feature as well. But if we look at the tongue and up closer, we have these taste papilla, and within the grooves here are the taste buds. And the taste buds hold these taste cells, and there's multiple types of taste cells. And it's very important to understand as we think about modifying taste, modifying sweet taste, that anything that is tasted has to go into this pore, which is about the diameter of a human hair, and it has to be dissolved in saliva and the mucus that is present in this taste pore. And we have different types of taste cells. We have cells that sense salt or sour or sweet or bitter. By and large, these taste qualities are sensed by different cells, different cell types. The other interesting thing is that taste cells are replaced about every 10 days. And so this provides the opportunity for the system to be shaped by experience. And we do know that there are changes in the density of different types of receptors, for instance, bitter receptors in particular, with experience. So if you are exposed to a high level of particular bitter compound, the amount of that receptor is downregulated. So this helps the system to be responsive to its environment. So as I said, for the most part, the cells are single modality, but we have learned that crosstalk can occur between these cells. And also the neurotransmitter for taste is the chemical ATP, which is the cellular form of energy. So when a cell is activated, that triggers a cascade of events within the cell, ultimately triggering the release of this ATP, which signals the taste nerves. Zooming in farther, I just wanted to make a couple of points about the receptor itself. So this is the sweet receptor. It's two 7-transmembrane domain helix receptors that form sort of a clamshell shape on the outside. But it's very interesting because it has a lot of different sites that different types of sweeteners interact with. So sugars, aspartame, some small molecule sweeteners interact with the outer part of the receptor. But there's other types of sweeteners like brazine or cyclamate that interact with parts of the receptor that are more into the membrane. So the cell membrane would go through here. And what that means is that these different types of sweeteners will have different kinetics or different onset and offset parameters. So the duration of the sweetness, the rapidity with which the peak sweetness occurs, and so forth, will all vary. And part of the reason for that is because they're binding to different places on the receptor. And it's a lot harder for something that's binding down here. It's going to take longer to probably trigger the sweetness, and it's going to take longer to decay afterwards. And I think, as you'll hear later, one of the concerns that people have is that these other alternative sweeteners don't have the same onset, peak, and offset that regular sugar does. So, we're not very good at fooling Mother Nature. We do know that our brain can distinguish caloric from non-caloric sweetener, even when the sensory experience is matched as best we can. And reward areas are differentially activated for different types of sweeteners. But this tells us that we know they're different, but it doesn't really tell us why. So to understand that why, it will help us to craft foods and diets that can satisfy our sweet tooth with less sugar and help to maybe devise ways to shape our preferences to accept less sweetness in our diet. So I'm going to talk a little bit now about the signals that are being triggered by these different types of sweeteners. So as I mentioned, this is the G protein coupled receptor for sugars. It's called T1R2 and T1R3. And all sweetness stimuli can act on this receptor. That receptor triggers G protein, which is coupled to various signaling cascades, resulting in an increase in calcium, opening of an ion channel, and ultimately, as I said, release of ATP. We know that removing this receptor in a mouse, for instance, eliminates the response to non-caloric sweeteners, but sugars can still be detected by these animals that lacked this receptor. So a number of years ago, some work was done to try to understand why. And what was learned was, what was learned was that there's actually two pathways for sensing sugars. We have this pathway that I just showed you, which is the G protein coupled receptors. And then there's also in taste cells, transporters. So there's a glucose transporter and a sodium dependent glucose transporter. And these transporters are present on the surface of taste cells and transport glucose into the cell, which is then used to make ATP. That ATP then closes a potassium channel, which results in a change in the membrane polarization, and again can result in further signaling in the cell. So glucose itself is somewhat sweet, but it's not terribly sweet. But you might ask, well, what about sucrose? Can sucrose be sensed by this pathway? And it turns out that there's also amylase enzymes and sucrase enzymes, disaccharidase enzymes on the surface of taste cells. And these enzymes can break down starch and disaccharides so that you do have additional presence of glucose monosaccharides at the level of the taste pore. And that is contributing to the ability to detect caloric sugars in the absence of this T1R pathway. And this was initially demonstrated in rodent species. And recently, researchers at Monell took advantage of the fact that this drug Florazin blocks this SGLT pathway, this transporter, to see whether it contributed to human perception as well. And I'm just showing you one of the pieces of data from this publication by Breslin and colleagues in 2021, showing that when you add Florazin to a glucose solution, the threshold, the detection threshold, is dramatically increased. So this is just with water. When you add sodium, because this is a sodium dependent pathway, the threshold goes down, so you actually can detect a lower concentration slightly. But when you block that pathway, the ability to detect glucose is much poorer. So this this effect is not seen with fructose. Fructose acts only on the G protein coupled pathway. It is not transported by this glucose transporter. So this shows that even in humans, this pathway does play a role in perception of glucose and other sugars that can be converted to glucose, like sucrose. So another important aspect of taste is these multisensory interactions. And we know that all of the different taste modalities can interact. Anybody who's cooked can tell you that. Salt has a direct ability to suppress certain kinds of bitterness. Sweet and bitter interact. Salt also enhances sweetness, and so forth. And there's also odor and taste interactions. Work has shown that with strawberry aroma, things taste sweeter, and vanilla aroma can also enhance sweetness perception. So these are odors that are associated with sweet taste, and so the brain kind of puts that information together and enhances the sweet perception. But what we've also learned recently is that there's odor receptors on taste cells. And this, again, I'm just showing one example of a particular odor receptor that is being expressed. This is in a mouse taste bud using green fluorescent protein that is present on many taste cells in the taste bud. So there's also the potential for odors to directly influence the function of taste cells, although we don't know yet exactly how this interaction works. So basically, these interaction effects are mediated by interactions in the brain and in the periphery. And as you'll hear in the next presentation, as people are experiencing sweet foods, whether it's a beverage or a pudding or what have you, their perception and their hedonic response to that food is going to be shaped by factors that are arising in the periphery. So the context of the sugar makes a big difference, and as well as the prior experience with those flavors and how much has the brain associated a particular odor with sweetness. And if there's odors that are congruent or consistent with sweetness, that will be a better environment than if the odors are inconsistent with their previous experience of sweet compounds. So just a couple of comments on how type 1 diabetes alters taste and smell. And this is an area that has not been studied as extensively as it probably should be. But we do know that insulin is a growth factor for cellular replacement in both taste and smell systems. There's in type 1 diabetics, there's fewer taste papilla. The individuals tend to be less sensitive to bitterness and sourness. And in some studies, they've been reported an increased expression of sweet receptors. There's also reports of impaired olfactory function. So if your sense of smell is affected, is less functional, you obviously will be getting less of those multisensory interactions than somebody with a normally functioning sense of smell. And diabetics tend to have greater susceptibility to smell and taste loss from viral infection. And so this is another complicating feature when you're trying to design foods and diets for people with type 1 diabetes. Another factor to consider is that there can be hyposalivation, which would carry with it less of those enzymes because the alpha amylase is present in starch, and it's also present on the taste cells in saliva, and other types of enzymes and degradation and binding factors as well. There's less intraoral distribution of taste stimuli. So as I mentioned, those taste pores are very small. Everything that's tasted has to be dissolved and get into those taste pores. So change in taste in saliva composition or quantity can affect the delivery of taste stimuli to the taste receptors. And so the impact will vary in different food contexts. So a few take-home points. There is an evolutionary inborn drive for sources of energy, and shifting preference for sweetness is going to be more difficult than it has been for salt. And the research that is going on, there's a large study in Europe that is currently in progress, and a large study that will be getting started in the US that's a joint effort between Monell and the US Department of Agriculture that was delayed by COVID. And so those data will be coming along, but unfortunately they've been quite a lot delayed by COVID. There's a second pathway in taste cells that's not activated by non-caloric sweeteners. And this neural signal resulting from ATP synthesis by glucose is not tasted per se, but it may contribute to the neural recognition of sweetness. So the brain is getting information from this second pathway that's allowing it to discriminate between a non-caloric and a caloric sweetener. And taste and smell interact both centrally and peripherally. And so it's important to really not think about sweet taste in isolation, or any taste for that matter, because it happens in a context, and these other senses have a big role to play in how we respond to that taste quality. And type 1 diabetes alters the taste and smell systems in fundamental ways, and this will impact flavor perception. And I think we need to know a lot more about the specific effects and the effects of different types of control of the diabetes and age superimposed on that, and so forth. But right now, we can say that the flavor world of type 1 diabetics is certainly not the same as the flavor world of non-diabetic populations. So thank you. That's my presentation on the basics of taste. I would be happy to look forward to answering some questions.
Thank you for this excellent update on the state of the science, Dr. Rawson. We're going to shift quickly to our second presenter. Please enter your questions into the appropriate space. Our second presenter will be Dr. Halis Akturk. Dr. Akturk is an assistant professor of medicine and pediatrics at the University of Colorado at the Barbara Davis Center for Diabetes. Dr. Akturk is founder of the Diabetes Dialogue, the annual diabetes uptake and diabetes technology workshop for nationwide endocrinology fellows. He has been a speaker for the diabetes technologies and newer therapeutics in type 1 diabetes at multiple national and international meetings, and we'll be sharing some new data exploring the relationship between the use of low-calorie sweeteners and the quality of life. Dr. Akturk?
Thank you very much. Can you see in only my screen, one screen, or?
You might want to swap your displays, Dr. Akturk.
Okay. How about
Perfect.
right now?
Yep.
Okay. Thank you very much. So today my talk will be about assessing the relationship between low-calorie sweetener use and the quality of life measures in adults with type 1 diabetes, and I will discuss one of the studies that we did recently. So this research was supported by the IAFNS. So a little bit about background, then less than a third of adults with type 1 diabetes in the US meet the ADA goals for glycemic control, which means that how many of them are less than hemoglobin A1C 7. Unfortunately, despite all the advancements in the insulins and the diabetes technologies, still we cannot achieve an hemoglobin A1C less than 7 in majority of the people. And you know that the type 1 diabetes requires lifelong insulin. So we need to find some other ways to decrease their blood sugar and increase their quality of life. And the amount of carbohydrate load negatively affects postprandial glycemia, and uncontrolled diabetes has a negative effect of quality of life and can lead to complications. So use of low-calorie sweeteners, we hypothesize that could reduce carbohydrate intake and improve glycemic control, which should be, but we check that if there is any effect on the quality of life. So in our study, this is a single center study in the Barbara Davis Center, and we have about 8,000 patients with type 1 diabetes, including the adults and peds, but this study is only for adults, which is about, we have 3,000 to 4,000 every year see patients with type 1 diabetes. So we did a cross-sectional study. It's a survey-based online questionnaire study. And we recruited 500 adults with type 1 diabetes between age 18 to 65. And we used the HIPAA-compliant web-based application REDCap, which most of the universities are using for research. So the study design is cross-sectional. So it means that we have a hypothesis. The low-calorie sweeteners use may improve the quality of life, and we are testing that hypothesis. But it's for an association, because in the cross-sectional studies, it cannot show the causality, and association between the exposure and outcome can be bidirectional. So it's not only going to one way, it can be going to the other way too. So the temporality of the exposure and outcome cannot be ascertained in these cross-sectional studies. So the methods, we used a lot of questionnaires, and we send this questionnaire to our patients' emails. And there is one questionnaire was about low calorie sweetener specific questionnaire. And there were questions about their low calorie sweetener use and how much they know about it. All other questionnaires are validated questionnaires, which is the Diabetes Self-Management Questionnaires, DSMQ, and Harvard University Food Frequency Questionnaire, FFQ, an audit of diabetes-dependent quality of life at Quality of Life Questionnaire, and type 1 diabetes and life, it's the title, and food-related quality of life. So the others are the validated questionnaires have been used in many quality of life studies in type 1 diabetes. So we examined overall characteristic of study participants. We divided the cohort into low-calorie sweetener use in the last month, yes or no. And we looked at the differences in demographics, glucose control and management, and quality of life metrics. So we asked a lot of questions to highlight some of them. And we asked, have you ever heard about low-calorie sweeteners? And 99% of them said yes. Have you ever used low-calorie sweeteners? 90% said yes. And have you used low-calorie sweeteners in last month? And 68% of them used at least once the low-calorie sweeteners in last month. So we also asked which low-calorie sweeteners has been preferred choice, and 41% of them said most of them tried aspartame and they prefer. And the second one was with 31% with stevia, and the sucralose, saccharin, and others, monk fruit were coming the other short answers. How have you used low-calorie sweeteners? And most of them are using when they are something drinking, and sometimes they are using in baking goods. But this was a multiple choice question, so 95% of them at least used the low-calorie sweeteners in their drinks. So how about the low-calorie sweetener and the diabetes control? And do low-calorie sweeteners help you to have better glucose control, such as better hemoglobin A1C? Time in range means that, if they are using a continuous glucose monitor, are they in range, in the normal range for the blood sugars or average blood glucose? And 73 of them said yes. And so that's important, because I think 73% of the people with type 1 diabetes say that, the low-calorie sweeteners, when they use, they have a better control. So we asked if they have a sweet tooth, and most of them said yes. And we asked, do you have any problems with using low-calorie sweeteners? 81% said no. And do you have any health concerns about low-calorie sweeteners use? 63% of them said no. So whoever said yes in these questionnaires, and then we ask them why with an open-ended question. So the next slide is going to discuss about the people that have some concerns that they said yes. So, and we ask why. So, and the next slide will be focusing on that. But the majority of the people, 81%, and said that they don't have a problem, and 63% said they don't have any health problems. So the people that, again, this small portion that say that yes, they have some concerns, and or they have some problems to use it. The most common problem that they were raising, that the taste. So, and they say, and again, this is an open-ended question, is accumulated the answers. And the second one was health-related concerns, and the third one was cost. And we focused on the health concerns. We asked for the say that health concerns, and we just asked them again, what do you mean? So almost half of them are just generally, oh, maybe there are some health effects that we don't know, or it may not be good for me. So there is not specific answers. Most of them you can see in this green pie chart. And the almost quarter of them mentioned that, oh, they may cause cancer. I heard it, I Googled, or somebody told me. And other up to 20% or so persons say that they are artificial sweeteners per se, even though we don't call like that. So they say they are artificial sweeteners. So the word artificial is, I don't like it. So it should be some chemicals in it. so that may be harmful. And a small portion of them said that, oh, I heard aspartame, can be harmful, or some of them still has some blood sugar concerns, a very small amount, or it may increase my blood sugar, or it may increase my weight, or it may change my gut microbiota or anything like that, because some of them has celiac disease. So the majority was concerns about that, generally non-specific health concerns. So we asked them, have you ever received any recommendations not to use low-calorie sweeteners from a healthcare professional? And 92% of them said no. So it's that all about that, the people that don't want to take it, actually, they are not getting this information from a healthcare provider. So they are just Googling or looking at some things, and they are just assuming or anything like that. So it's all about that, the interactions with different people or using Google or anything like that, but not the healthcare providers. So you are currently using low-calorie sweeteners. Do you think low-calorie sweeteners have changed your quality of life? For the people that are using, and 53% of them said yes. These are the users, and 53% of them said yes. And we ask in terms of what? Is it making it worse, or is it making it better? And 92% of them said better. So the low-calorie sweeteners users, most of them, they think that their quality of life is improving, and most of them are happy about it. So we ask the same question for the non-users. You are not using low-calorie sweeteners. Do you think low-calorie sweeteners would change your quality of life? Again, this is a hypothetical question because they are not using anyway. And 88% of them said they are not. It's not going to happen. So we asked them if it would change, it would make it better or worse, and 73 of them were thinking it would make it worse. So these are the people that are mentioned that they have some concerns for different reasons. They are already showed in the previous slide. So we asked, have you ever tried to cut down your sugar intake? And 75% of them said yes. And again, remember that they all have type 1 diabetes. There's a correlation how much carb you get, and your blood sugars will be higher, whatever you do. We have advanced technologies right now, smart pumps, insulin delivery systems, pens, but everything is really depend on what you eat at the end of the day. And if yes, what was the purpose? Have you ever tried to cut down the sugar, and why are they cutting the sugar? 81% of them said to have a better glycemic control. So 81% of them are really focused on to decrease their blood sugar. And 60% says to lose weight, and 33% says to increase the quality of life. So the people's priorities are different in type 1 diabetes. We see that they want to focus on to decrease their blood sugar first, which will eventually increase their quality of life too. And they want to lose weight, which will eventually increase their quality of life, but they are not thinking the first one, oh, it will increase my quality of life, but doing everything will increase their quality of life anyway. So the demographics of our population, that the average age was 36-year-old, and 31% of them were male. And we see that, even though we have a well-balanced, female-male ratio in our patients, in any survey study that published, you can see that there is a significant higher response by females. And the race-wise, I live in Colorado, so it's the Colorado population, mostly 92% of them are white. And so the education-wise, graduate or professions, 26%, college graduate were 42%, and some colleges, the 24%. These are the higher numbers. Insurance-wise, 82% of them are private insurance, and 17 of them are Medicaid, Medicare, kind of government-based insurances, and 12% of them were smokers. So as I said, we divided these people that this recent 1 month use of low-calorie sweetener that they are using or they are not using it, and this 360 patients versus 172 people. And then you see that the age and diabetes duration were different from the baseline. the people that are using the low-calorie sweeteners are actually a little bit older, statistically significant, and their diabetes duration is higher, statistically significant. So it shows that the people are actually have more diabetes are actually try to use the low-calorie sweeteners. All other everything, they were not significant in the baseline. So history of complications by low-calorie sweetener use, and if you look at that, that's the only difference was the hypertension at the baseline. So the other things were not significant. So the people that decided to use the low-calorie sweeteners had higher blood pressure previously, but before then the other complications are the same. But if you look at the any complication rate was also significant, and the people are recently using, they have higher complications before they have been starting using it. So how about the glucose management by low-calorie sweetener use? And this has been adjusted for age, sex, diabetes duration, and complications. And if you see that there was no difference between that users in the recent users and not users for their hemoglobin A1C, DSMQ, which is the Diabetes Self-Management Questionnaire score, and DSMQ also has sub scores that I showed that the diet has no difference. There was the only difference was the only in the physical activity, which the non-users has a slightly better quality of life. And in the DSMQ score, again, we use some other questionnaires to assess the quality of life, and I will show in the next slide the other questionnaire results. So the other questionnaires that we used, 3 other questionnaires, had similar results actually. The AD quality of life score, Untitled score, and the food-related quality of life, and there were no difference between the current use and no current use for the quality of life. And these again adjusted for age, sex, diabetes duration, daily breakfast, eating outside of the home, trying to lose weight, and complications, which pretty much adjust for everything else. And to summarize my part, the low-calorie sweetener use, most adults with type 1 diabetes in our survey were aware of and had used low-calorie sweetener, with over 2/3 of them using low-calorie sweetener in the past month. The most common use reason was using in the drinks. Most people were using low-calorie sweeteners to meet glycemic targets and improve their quality of life. And to emphasize again, these people are lifelong insulin dependent. So their major focus is to decrease their blood sugar, to decrease the long-term diabetes related complications, and eventually quality of life comes with better blood sugar. And people with type 1 diabetes who reported using low-calorie sweetener in the last month were older and had longer diabetes duration and more complications than those who reported no recent low-calorie sweeteners use. So it tells you that the people have, obviously, if they have more complications, longer duration of diabetes, it means that they have worse, diabetes control, to relatively speaking, accumulating in the long term retrospectively. So they are trying to find some ways to decrease their carb intake, calorie intake, and decrease their blood sugars, and eventually trying to improve their quality of life. And recent low-calorie sweeteners users were similar to non-similar in terms of the hemoglobin A1C, diabetes self-management, but had lower scores on physical activity, diabetes management. Again, we only check the people's hemoglobin A1C as a snapshot during the study. So remember that hemoglobin A1C is a tool that only shows the last 3 months of your average. But if somebody already has a lot of complications, it means that their hemoglobin A1C have been higher compared to the other person. So this second and third phrase actually the same, so it doesn't contradict itself. So I just wanted to clarify that part. The lower quality of life score and on the tidal questionnaire among recent users of low-calorie sweetener was attenuated to non-significant when adjusted for diabetes complications and poor dietary practices. So the conclusions of my talk, the take-home points are: adults with type 1 diabetes in this survey reported a high degree of knowledge and use of low-calorie sweeteners, and there were no differences in current reported glycemic control and quality of life between recent low-calorie sweeteners users versus non-users. Diabetes management was similar between recent low-calorie sweeteners users and non-users, except for physical activity. That was a little bit better in the non-users side. Low-calorie sweetener users report benefits for glycemic control and quality of life. Education is needed about the low-calorie sweeteners and related health concerns. As I mentioned, they had a lot of concerns for the small portion of the people that are not using. And further research is needed to understand quality of life and low-calorie sweeteners relationship, and probably to address their concerns. So when I conclude my talk, I would like to thank to my colleagues here, the Janet Bergen and Viral Shah, and for the IAFNS for the sponsoring our study. Thank you very much. And I will be happy to answer your questions.
Thank you for sharing that very interesting data on the novel connection between sweet and quality of life. We can now shift to addressing some of the many questions from our audience. So one of the questions that we would have for Dr. Akturk would be that you've talked about the need for trying to determine the difference between using the low and non-calorie sweeteners and the quality of life. What might be your next step to try and tease apart this interest?
Sure, I think we need more studies to see this, if there is any association or any causality relationship in a prospective way. We can just make some studies in the people with type 1 diabetes, and we can supply the low-calorie sweeteners, and then in a prospectively long term we can assess a better way to their glycemic control and also their quality of life with the validated questionnaires. But regardless, if these people achieve better glycemic control, their quality of life will improve. So there are many studies show that however you just get a better glycemic control, their quality of life improves. So even indirectly, that if you can just get a better glycemic control, and they can just eat, because anything is dependent in type 1 diabetes, how much carb you get. So we are not advocating to eat a low carb either, because it may trigger a diabetic ketoacidosis in type 1 diabetes, but it should be not too much. So I think if some of the replacements can be done with a different way, and it will be helpful. And the other part is that we need to show these people that they have a lot of concerns. You see that more than 90% of them mentioned that one of the healthcare providers never told me that don't use it. So why you just don't use it? Because I heard from this, I heard from that, or I read from this, and these kind of things. We need to address these people's concerns. I think that's the part that they need more education, and they need to get the more scientific information.
Thank you for that. And a question for Dr. Rawson. Do low and no calorie sweeteners uniquely drive a preference for sweet because they have a very high sweetness intensity? You might have touched on this a bit in your talk, but perhaps we could review it.
Sure. So there's evidence that a high sweet diet, regardless of the cause of or the source of the sweetness, can increase the pleasantness of sweetness. So a higher concentration becomes pleasant. But there really isn't evidence that non-caloric sweeteners do this in a way that's different from sweetness generally. I think there's certainly more research to be done in this area. And the fact that as we've learned about this second pathway that is only sensitive to glucose monosaccharides, suggests that there's probably more to sweetness than we understand at a molecular level, at a cellular level, and that you know, having a product or a food that has only non-caloric sweetness and no caloric sweetener may be altering the brain, how the brain is learning about this food and the subsequent processing of that information and so forth. So, you know, I would say at the moment, it's really, it's not that the sweeteners are unique per se. And if anything, most of those sweeteners do have off tastes or other tastes that are really not necessarily pleasant. Sometimes it's bitter or lingering or different things. So I would say at the present time, there's no evidence to support that.
Thank you. But we have time for a few more questions. In my box, I don't see any additional questions. Marie, I'm wondering if there are questions that I'm not seeing.
Yeah, and if you're in the document, you can scroll up to the top.
Mm-hmm.
There are several questions there. Okay. So for example,
I see them, yeah.
Okay.
So for both presenters, our listeners or participants are interested to know what do we know about the microbiome changes that might play into the changes of taste preference? Are there negative effects of low-calorie sweeteners on the gut microbiome? Do either of you have any comments?
Yeah, I'm not an expert on the gut microbiome, unfortunately. I would say there's also an oral microbiome. I do know that there's studies showing that these things change, you know, in the presence as diets change, the microbiomes change very dramatically. One thing that we don't understand, and is another really important area, I think, to think about, is how oral microbiome in particular influences our sense of taste. We do know that some of the bacteria release different types of chemical compounds, some of which are bitter, are perceived as bitter, but others which may not be perceived as taste stimuli necessarily, but act on specialized sensory cells called solitary chemosensory cells. And these cells are very important in regulating the immune response in the oral cavity and in the gut. And so and also in kind of monitoring the bacterial populations. So I think that's a really interesting and a great question. But I would say that in terms of understanding how the microbiome alters taste, we just don't really have the data to talk about yet. Thank you.
Okay, thank you. So someone asked about self-report. I think all the data that you presented, Dr. Achter, were self-report data. Isn't that correct?
Correct.
I don't know if there's any way to get it, something that isn't, you know, I mean, in further studies.
I mean, most of the time the self-reported results are very, you know, validated in type 1 diabetes because these people are, you know, seeing their physician every 3 months. Their hemoglobin A1C has been checked every 3 months. You know, type 1 diabetes is a lifelong, you know, issue. So they know if they have a diabetes-related complications or not right away. So, you know, it's not something like, you know, you may not know if unless you have not been to your physician like many years, which is, you know, not going to be physically possible. So there are studies show that, you know, self-reported studies are very valid in type 1 diabetes field, especially even for their hemoglobin A1C or anything like that, because that's the most important number in their life to know. So.
Dr. Achter, there's a question here about, do you know if those who do not use the low or non-caloric sweeteners have a higher sugar intake or higher intake of sugars?
Yeah, we didn't look at specifically for these people are if they are, you know, and this can be a sub-analysis we can do, but we haven't looked at yet if there is any correlation or not. That we just looked at the whole population for their how much they are sugar intake in terms of if they have a sweet tooth or not, or if they prefer sugar in their food or not. So,
Thank you. Another question for Dr. Rawson. Are the flavor and taste disorders that you have seen in patients with diabetes the same for type 1 and type 2 diabetes?
No, it is a bit different in type 1 versus type 2. Type 2, they tend, I mean, provided, of course, I mean, obviously this depends a lot on the environment, the degree of control, and the diet and so forth. But the loss of taste papilla and taste cells is not typically seen in type 2 diabetes. So there are some subtle differences in the gene expression in type 2 diabetes that are, it's not quite clear how much of that is the diabetes, how much is the diet experience. But I would say that the biggest difference is that the lack of insulin in the oral cavity and in the taste bud environment is impairing the ability of those taste cells to regenerate, because it is a growth factor for taste cells.
Thank you. I think we have time maybe for one more question. Dr. Rawson, can you explain how the taste buds were impacted by COVID and why they weren't recycled in the 10-day span, or if they were, but were modified?
Yeah, I was hoping somebody would ask about COVID. Yeah, so it's really, so that the ACE2 receptor that the virus latches onto is present not only on the mature taste cells, but also on the stem cells that develop into the mature taste cells. And so the virus was taking out not only the mature cells, but also the cells that regenerate to create new taste cells. And so that's why the the recovery could be delayed, although generally it was for taste, that the sweet, salty, sour, bitter, when people lost their sense of taste, it was very sudden, but it did tend to recover within a few weeks. So but it didn't necessarily recover in 10 days because the stem cell population was also affected. For the sense of smell, the delay in recovery is more complicated, and we're still trying to really understand why in some people, 2 years after they've lost their smell, they still have no sense of smell. And so that's more, but again, there's changes in the olfactory system, in the ability of those cells to make new olfactory neurons. But the regeneration of the olfactory neurons requires that they not only make new mature cells, but those cells have to send axons back to the brain. The axons have to get to the right spot in the olfactory bulb and make the right connections in order for the sense of smell to be fully recovered. And that's a longer process, and age affects that process. Other kinds of conditions affect that process as well.
Thank you very much. Well, that brings us to the end of our time today. A reminder that access to the CEU certificate will be provided by email after the close of the webinar. Thank you to everyone that participated and offered great questions. And an even bigger thank you to our speakers, Dr. Rawson and Dr. Aktürk, for taking time to share your insights with us. If you are interested in more science-based CEU-approved programming, visit the IAFNS website under Continuing Education. Thanks, everyone, and have a great afternoon.
Thank you. It's great to be here.